is CORDIScovery. Hello and welcome to this episode of CORDIScovery with me, Abigail Acton. Biodiversity, a key barometer of the planet's health, is under threat from climate change and pressures on natural resources. But it's not all grim news. Science-based conservation can step in and halt a decline. In a paper published last year in the journal Science, researchers report the results of a global meta-analysis of 186 studies comparing changes in biodiversity over time. They looked at areas in which conservation had taken place and those in which there had been no action. Showing that biodiversity levels were clearly impacted. They concluded that in 2/3 of cases, conservation either improved the state of biodiversity or at least slowed declines. Today we'll journey through rainforests, air and sea to hear about the latest research funded by the EU that's gathering the data we need to make conservation effective. We'll be looking at how parasites are undermining the nutrition that feeds the sea, krill. We're considering what the past can teach us about the current state of migratory waterbirds, and we'll be asking what climate change is doing to our rainforests. Based at the French National Research Institute for Sustainable Development, Claire Fortunel is co-leader of a group examining the dynamics of tropical forests at the Joint Research Unit for Botany and Modelling of Plant Architecture and Vegetation in Montpellier. She is particularly interested in understanding tropical forest diversity and functioning and how these ecosystems will respond to global climate change. Hello, Claire.
Hello, it's great to be here.
Very nice to have you. Ada Álvarez-Manzaneda is a fellow at the Department of Ecology at the University of Granada in Spain. She is especially passionate about using paleoecological approaches to support waterbird conservation, uncovering past environmental patterns to guide future protection efforts. Hello, Ada.
Hi, how are you?
Very well, thank you. Alison Cleary is a molecular ecologist at the British Antarctic Survey. She is keen to improve our understanding of polar marine food webs and how the interactions between organisms shape our ecosystems. Hello, Alison.
Hello, Abigail.
Claire, I'm going to turn to you first. The Trop-Dem Trait Project wanted to improve our understanding of how the interplay of species traits, tree size, climate, soil, and interactions between species impacts tree growth and mortality rates in tropical forests. So, Claire, rainforests are vital for the health of our planet and rich in biodiversity. But can you give us some examples of how healthy rainforests benefit us all?
Of course. I'd like to invite you to picture yourself in a tropical forest. You can hear the many sounds surrounding you, from the howling monkey to the singing birds to the chirping insects. Now, look at the trees. They can reach up to 50 meters high, and as such, they're structuring the layers of the forest from the canopy to the understory. And this can influence how trees interact with each other, but also the many species that they can host. And this includes the macrofauna, such as the mammals, the birds, but also the microfauna and the microbes that we think less about. And estimating the number of trees is still a very active field of research. And in parallel, there are ongoing efforts to better estimate the diversity of the other organisms living in these tropical forests. For now, what we know is that we only know the very tip of this green iceberg of diversity, and they are providing essential services to human population, not only at the local scale, but also globally.
Excellent. That's a lovely description. And thank you for painting such a beautiful picture. I could almost hear the parrots. So that's fascinating. But we know that rainforests are threatened by exploitation, mining, logging, and clearance for farm and so on. You were interested in the stresses caused by climate change, though. So how did you go about assessing that?
Oh, we still have a limited understanding of how tropical trees live and die. But as tropical forests are facing climate change, we need to clarify how these trees will respond to climate extremes in particular, if we want to better protect them. And to address this question, we studied the variation in tree growth in response to climate anomalies. And these anomalies can be seen as unusually high heat or water stress. And to test our hypothesis, we used a modeling approach where we can track the effect of different climate stressors on tree growth. And thanks to the many collaborators who have collected tree demography data over decades, which means over 50 years, we were able to compare tree growth response to these climate anomalies across South America, Africa, Southeast Asia, and Oceania.
So could I just ask you, when you talk about the anomalies, obviously it's fluctuations, extreme fluctuations in weather patterns. You were talking about the collected data from decades ago. What form did that data take?
It looks like census data, so people going in the field in permanent what we call forest plots, where every stem that has a certain size, so a certain diameter, is positioned and monitored. So we have their diameter that is measured at regular intervals from which we can measure growth increments.
Oh, perfect. And then you can tie that in to weather patterns and the data that you have and compare the two together. Excellent. That's excellent. Could you carry on and tell me a little bit more about your methodology? What else did you do?
So we needed to evaluate also these climate anomalies. So for that, we had long discussions with climatologists to better understand what climate anomalies actually are and tie that to the biology of the trees. So what do trees experience? And we came up with several ways of calculating how this different heat and water stress could impact directly or indirectly tree growth.
Okay, super. And what did you see?
So what we found is we can summarize it in two main points. The first one is that across the tropics, we found that tree growth declined with increasing warmer and drier conditions. And that means that tropical forests worldwide will be similarly impacted by climate change. And this stresses out the urgency of curbing our global carbon emissions if we want to protect these areas. And second, we took a closer look at where exactly the over 100 sites that we studied across the tropics were positioned along a temperature gradient. And what we found is that warmer forests were more sensitive to climate change than colder forests. And this suggests that these areas might call for more attention from our part if we want to protect them.
Because they're more sensitive, indeed, as you say. Did you find whether other tree species were taking the place of the ones that were dying due to stresses or that were becoming weaker? In other words, you're describing the trees as getting smaller in diameter and the growth being less marked. That must mean that there's more space for other species to grow. What did you find about the equilibrium between the shrinking and the space left behind, the vacuum maybe?
This is asking a broader question of forest dynamics as a whole. So not only how trees grow, but also how they die. What we find is that overall, over time, we're losing the species that are adapted to wetter climates at the benefits of those that are better adapted to these warmer and drier conditions. And these are really long-term changes because mortality rates in tropical forests are around 1 or 2%. So very few trees actually die. There's very little room for trees that are trying to recruit. But still, that means that there could be, with only 1 or 2%, a lot of changes in the carbon that is being sequestered in these tropical systems.
Yes, and also I imagine if the species of the trees change, then do the dependent species also change? All the organisms that they host, presumably they're not necessarily the same.
This is a very hot topic of research, as I was mentioning, because we're still looking at estimating the global diversity across all these trophic levels. We're still very much at the beginning of understanding what is going on. But yes, there can be cascading effects across trophic food webs, and this can have profound impacts on this tropical diversity and the functioning of these ecosystems.
Super. Thank you so much, Claire. That was really well explained. I really appreciate that. Claire, studying trees must be quite challenging, given that, as you said yourself, you've got 50 years worth of data. It's a slow process. The trees might outlast us or the researchers that are doing the work. So how do you try and get an idea in a way that sort of speeds up the process so you can comprehend it a little bit more, I would almost say quickly?
This is a huge challenge for ecologists, especially working with trees that can live for centuries. One idea that we are currently pursuing is to test if characteristics of trees that we can measure today, such as in their leaves, their stems, or their roots, can inform us on their long-term response. So not only growth, mortality, but also how they reproduce. For instance, does the way a leaf handles water stress can help us capture how trees will respond to extreme drought?
Okay, that's great. So it's like taking a snapshot of one particular part of the tree to see if you can extrapolate how it's going to respond more generally. Excellent. Thank you so much, Claire. Thank you very much.
Thank you.
Does anyone have anything else to add or any observations to make? Yes, Alison.
I had a question. Talking about all of these longer term changes as the temperature warms, does your data tell you anything about how reversible they would be? Like if we managed to change the trend of climate change and bring temperatures back down, would those trees come back, or is it moving towards a tipping point where it would end up a very different ecosystem?
This is very much an open question, especially in tropical systems. I didn't mention it earlier, but we're also looking not only at tropical rainforests, but other tropical woody systems, including savannas. So what we have found so far is that looking at how resilient tropical trees are to the compounded effects of drought, they are less resilient than species that are found in drier ecosystems. So it might be difficult for them to really come back to a pre-extreme drought state.
Perfect. That's fine. That's excellent. Alison, over to you now. The Paracryl project used new molecular tools to reveal a much higher than expected diversity and prevalence of parasitic infections in krill. So can you tell us about krill? What are they and why do they matter, Alison?
Yeah, so krill are small crustaceans. So they're animals related to shrimp and lobsters. And I focus on Antarctic krill. So when they're full grown, they're about the size of a crayon. And these are really important parts of Southern Ocean food webs because they're prey for the fish, seals, penguins, flying birds, sort of all of the charismatic Antarctic wildlife. And they're also important in carbon cycling. So when they eat tiny plants near the surface of the water and then poop, basically, that sinks to the sea floor, that transports carbon down and away from the atmosphere. And they're also the target of a growing commercial fishery. So they have an economic role to play as well. So really important animals.
And yeah, clearly so. But what made them important to you? Why did you decide to study krill?
They're just fascinating and incredibly beautiful little animals with just intricate, elaborately feathered appendages, so well adapted to their habitat. You know, they can feed on the ice or the sediment or the water, and they even bioluminesce. They're just amazing.
They kind of got everything, huh? Okay,
Yeah.
Excellent, super. So what did Paracryl find out about the relationships between parasites and their hosts?
Yeah, so we found quite a diverse range of parasites, and we found them in many of our krill. So we found single-celled parasites, things like ciliates, single cells with lots of hairs around them, as well as a group called Apicomplexa, which they look a bit like peanuts. And we found multicellular parasites, so mostly worms, nematodes, and trematodes. And some of these parasites were known to be common in krill. Some of them had never been reported from the Antarctic krill or had only been quite sporadically reported.
And why does it matter?
So we were looking, that was our second question, was do these parasites actually impact the krill? Because if it has a parasite, but it doesn't really care, then why bother? So we looked at the krill, whether it affected how they reproduced and how they grow. And we found that krill, which had a lot of parasites, produced eggs that had less nutrients, so carbon and nitrogen in them. And how much nutrients are in an egg, that's how much resources that embryo has to grow up until it's big enough to fend for itself. So if less resources, that's less chance of growing up to be a successful adult. We also found that krill, which had a lot of parasites, grew more slowly. So that means it takes them more time to be big enough to be a good prey for a penguin, for example, and that they'll have a different role in these carbon sequestration.
Okay, obviously that's pretty fundamental. But how did you go about finding out about these parasites? I'm kind of curious about the method here because I'm seeing, size of a crayon is actually quite big, but I'm seeing very small little krill kind of growing up or even just sort of in the egg. And yeah, how did you work out what was actually inside them? What did you do to find all this out, Alison? Tell us more.
Yeah, so first we had to catch the krill. So that was in Antarctica with several collaborators helping us on this project. Then we kept krill in this big aquarium. We call it the Krill Hotel.
Excellent.
It's about the size of a bathtub. And within that hotel, we have hotel rooms, which are small jars with little perforations so that the water can flow through them. So there's one krill in each hotel room. And we check on this multiple times a day. So that lets us measure things like, are the krill laying eggs? And what can we learn about the eggs? And also, how fast the krill are growing. So unlike Claire's trees, our krill can't just grow slowly and gradually. Because they have exoskeletons that don't stretch, they can only grow by shedding the exoskeleton and then making a new, bigger one. So we look for when they've shed an exoskeleton, because then you can measure the size of the exoskeleton and the size of the new krill.
Right. It's quite convenient, actually. Like shedding a coat.
Exactly.
Yeah.
So then we take all of our krill back to Cambridge, and we blend them up into sort of a krill smoothie and extract all the DNA from that. So then we can sequence the DNA, and we include a special custom protein that means that we don't sequence the krill itself. So that lets us see everything that's not a krill inside the krill. And you can compare those sequences to a database to figure out what all the parasites are within each individual. and then tie that back to things like which eggs did they lay and how fast did they grow.
Wonderfully explained. That's excellent. Thank you very much. Alison, obviously, as we were saying earlier, krill is a basic food source for so many animals within the ocean. But haven't krill always been subjected to parasites? Aren't parasites just part of krill daily life?
I think parasites have probably always been part of this food web. But one of the questions we're interested in is how might that change? So if changing conditions shift the balance between parasites and their hosts, how does that then affect the ecosystem? So we really wanted to sort of understand fundamentally these interactions between krill and their parasites so that we can build that into our understanding that we can use for management. Because of course, if we have fewer krill or the krill are taking longer to be size that they need to be prey, that's going to affect what's available for fish to feed on, for seals to feed on. The Antarctic feels like it's the end of the world, but things in the Antarctic do affect the rest of the planet. So, for example, humpback whales feed really heavily in the Antarctic in the summer, and then many of those migrate north. And Arctic terns are feeding on these Antarctic krill and then migrating north for the winter.
Thank you, Alison. That was beautifully explained. Thanks very much. I love the idea of a krill hotel. Does anyone have any comments or observations to make for Alison?
Yeah, I have a question.
Yes, Ada, what's your question?
So thank you, Alison. I would like to ask did you expect to have different results in other climatic environments? I don't know if the climate change, for example, is going to be beneficial or so for the parasites. I'm not sure.
I think that's something that we still don't really understand. Every organism has the temperatures where it functions best, both parasites and hosts. So as temperatures shift, that balance can shift, but it's complicated. It's not sort of a linear projection, and whole communities can be shifting. So it's definitely something that we're interested in, and we are working with krill from different areas around Antarctic to compare these areas, but definitely something to keep looking towards in the future.
Super. Thank you. Very good question, Ada. Thanks very much. And I'm going to turn to you now. Let's talk birds. Inland waters are recognised as important sentinels of climate change, and their sediments accumulate records of past environmental conditions. The PAST project studied the trends in environmental changes to improve our understanding of current challenges faced by migratory water birds. So can you tell us what paleoecological studies actually are, Ada, and what can they reveal?
Yeah, sure. I mean, we can define the paleoecology as the ecology of the past. So this science gives us information about how were the ecosystems in the past. And when these approaches are applied to aquatic environments, this is known as paleolimnology. Since we have to know that the sediment of the mud in the bottom of the lakes and ponds act as historical archives of the environmental conditions that happened in the past.
Excellent, wonderful. And how do you go about studying the sediments? What's the process to study the sediments? I'm just seeing a massive amount of mud. Please help me to understand better.
Yeah, sure. I mean, I'm focused on the study of the population, the changes that happened in the past in several water birds populations. So the first step is, of course, to sample this mud, this sediment from the bottom of the lake. So for that, we have to use something that is called sediment cord, that is a tube that helps us to retrieve the sediment from there, from the bottom. So after we get the sediment, the mud, we have to section it because we have to date this. We know that there is a lot of data there, a long time. So we want to know what was the different periods of time that are there. So we have to section it by using different intervals. This is going to depend on the resolution that we want to get. And after that, we have to freeze dry the sample that we get, the different intervals. And once that we have these samples, this sediment, this mud dried, we can now analyze it or process the samples for the different analyses that we are going to do.
Okay, and what did the analysis show you? Actually, what did you find when you went through that process?
Yeah, of course. I mean, the first step is to date the sediment. Sometimes the sediment is very disturbed, so it's like mixed in that, if that happens, it's impossible to date it because everything is mixed. But if the dating is good, we can analyze, for example, one of the main proxies that they used to use is the diatoms, that are some algae that can provide us with some information about past environmental conditions, such as the moisture of or nutrients presence. Okay.
Wonderful. So the presence of these little plants, I guess they're plants, yes? Little tiny plant species.
Yeah, kind of.
Kind of. Oh, that sounds like amazing.
They are plants, but they live in aquatic ecosystems.
Yes, sure. Absolutely. Yeah. I like it when the edges get blurry. So the presence of these little plants can tell you about the environment at that particular period, and you've dated it according to the way that you've managed the core. Can I ask you what you learned about the migratory bird populations by doing this?
Yeah, because I work also for knowing about birds or other, let's say, animals. We can use an isotope, that is the isotope nitrogen 15. This isotope is increasing by every trophic level. So by knowing the, let's say, concentration of this isotope, we know what kind of animals were there in the pond in this case. So by analyzing this nitrogen 15 and by comparing this with the information about the moisture that diatoms are giving to us, we can know what was happening in the past, because we can relate that with the date that we know for sure that these diatoms were present, or this nitrogen 15 was higher or lower.
It's wonderful how much this can reveal, Ada. How far did your project actually manage to look back? In other words, what's the sort of earliest data that you have?
Okay, we used to focus on the previous, the pre-industrial era, and we have been able to reconstruct bird populations longer than 700 years old.
Goodness me. Wow, that's a really good snapshot. And what are you actually seeing with regards to the changes? I mean, you mentioned pre-industrial and now. Can you compare? What changes are you seeing in the populations of these migratory birds? And what birds are we talking about, Ada, actually, for example?
Yeah, for example, we have been working on the eider nest by applying in the Arctic, in the Canadian Arctic. And that was the first time that this approach was done.
So you're looking at the eiders, right? Okay.
Yeah. Uh-huh. Yeah. So in this occasion, we were working with eider nests, and we saw that in the past, before having hunting regulations, these populations were declining. And after these hunting regulations that were done in the 21st century, they were recovering. So we know for sure that hunting regulations are very, very important for this kind of birds.
Excellent. So there we have an example of a conservation action that actually turns the situation around. Fantastic.
Yeah, for sure.
And what else can you see about populations? What else have you learned?
Yeah, we can know also if some climate change or environmental changes were happening at that time and how they affected to these bird populations.
And what did you see?
Yeah, for example, midwinter in Canada after the Little Ice Age era were beneficial for these birds because they were also, let's say, the ice cover was smallest or thinner, let's say. So they were farther than these other animals that they can eat them. And also they were growing more and more, and they increased their occupation of the nest.
Fantastic. Okay, that's super. And again, we're with the eiders here. That's wonderful. Okay. Any other results that really seem to you to be quite surprising or interesting that you would like to share with us?
Oh, yeah, sure. I mean, we worked also in another area in the Canadian Arctic as well. And we saw that after the human populations were living there, were moving around, the bird populations in that area were declining as well. Not only because of the hunting or something, because they were disturbing these birds. So, and also, of course, because of the pollutions and the lead, that is also bad for the birds.
Yeah, I mean, of course, you can measure that in the deposits easily, I would imagine, the presence of lead.
Mm-hmm.
That's fascinating. Thank you so much. So it's interesting to see how much the sediments can actually show us about what was going on before we were even beginning to make records. That's excellent. And then, of course, you can compare it with the dynamics now. Very good. Thank you so much. Does anyone have any comments or observations to make for Ada? Yes, Claire, what would you like to ask?
Excited. This is a really fascinating insight into long-term bird change population, like population changes.
Thank you.
I was wondering if you have limited information you can get from your sampling. If you could go back in time, what would you like most to learn? Like if you could measure something new.
Wow.
That's a lovely question.
Yeah, that is. I'm not sure. Maybe, yeah, for sure. The increase in the human population, how that affected to the different ecosystem that were around, and also for sure because we know that everything is connected. You know, Claire was talking about the trees, Alison was talking about the krill. So everything is connected, and I would like to try to link everything. Like, okay, we know that if there are no trees, some birds are going to decline for sure because they use them for nesting or as a shelter. So I would love to connect everything and how maybe we also have been beneficial or so for some kind of species, but on the other side, we were detrimental for other species. So I would love to, but it's very tricky for sure.
Oh, yeah, that's massively ambitious, but it's a wonderful goal. And I think everyone who gets involved in biodiversity, that's the fundamental goal, isn't it? To understand the interactions on a wider scale.
Yeah.
Wonderful. Well, listen, I want to thank the three of you for sharing your fascinating work. I'm so glad we managed to come at this from different elements, as it were. So that's great. Thank you very much for your time. You've explained your work very, very well.
Thank you.
Thank you for the invitation.
You're very welcome.
Thank you so much for these very exciting exchanges. I learned so much from you all.
We all did. Thank you. Goodbye.
Bye.
Take care. Bye-bye.
If you've enjoyed this podcast, follow us on Spotify and Apple Podcasts or whichever services you use, and check out the podcast homepage on the Cordis website. Subscribe to make sure the hottest research in EU-funded science isn't passing you by. And if you are enjoying listening, why not spread the word? In past episodes, we've explored how drones can help prevent fires on ferries and how babies perceive objects moving towards them on a screen. In our last 44 episodes, there'll be something there to tweak your curiosity. The Cordis website has articles and interviews that explore the results of research being conducted in a very broad range of domains and subjects. From fishing to fishing, there's something there for you. Maybe you're involved in a project or would like to apply for funding. Take a look at what others are doing in your domain. So come and check out the latest research that's revealing what makes our world tick. We're always happy to hear from you. Drop us a line, editorial@cordis.europa.eu. Until next time.